My name's Kaylee McCoy. I'm from the Academy of Notre Dame, and the name of my project is the effect of various mouthwashes on Streptococcus mutans. The question for my experiment is which type of mouthwash, Scope, Listerine Antiseptic, Act, or Crest Pro Health is most effective in killing the S. mutans bacterium. The S. mutans is a gram-positive, facultative anaerobic bacterium. It's heterotrophic, which means it must live off other organisms by either eating that organism or using the organism as a host. In the case of the S. mutans, the human oral cavity is the host. It's also widespread, inhibiting 30 to 60% of the total bacteria within the surface of human teeth, tongues, and cheeks. Finally, the S. mutans metabolizes sucrose to lactic acid, which is what causes the tooth enamel to decay when mixed with plaque. There are many different types of mouthwashes with various purposes. The first is breath-freshening mouthwashes. Their main purpose is to reduce the odor within the mouth, but not necessarily kill all of the bacteria within the mouth while doing so. It contains a low alcohol content. An example of a breath-freshening mouthwash is Scope. Next, there's antibacterial mouthwashes. Antibacterial mouthwashes' main purpose is to fight the bad breath, plaque, and gingivitis. They contain high alcohol contents, which can reduce the saliva within the mouth, but at the same time dries out the mouth, causing bad breath later on in the day. An example of an antibacterial mouthwash is Listerine Antiseptic. Next are fluoridated mouthwashes. Fluoridated mouthwashes are best for those who are susceptible to cavities because fluoride is proven to help prevent cavities. However, it does not prevent against gum disease. They have a low alcohol content, which means it's not specialized to kill the bacteria within the mouth. An example of a fluoridated mouthwash is Act. Lastly, alcohol-free mouthwash's main purpose is to rid the mouth of bacteria without depleting the saliva supply. An example of an alcohol-free mouthwash is Crest Pro Health. My hypothesis for this experiment is if Scope, Listerine Antiseptic, Act, Crest Pro Health, and the commercial saline, which is the control, are tested, then Listerine Antiseptic will be the most effective in killing the S. mutans. This is because Listerine is an antiseptic mouthwash. Research has shown that antiseptics have the sole purpose of killing bacteria within the mouth. Meanwhile, other mouthwashes are often produced just to mask bad breath. In addition, Listerine is one of the few mouthwashes to earn the ADA seal of acceptance for effectively reducing gum disease and plaque. There are many different materials used for my experiment. First was the different types of mouthwash. Breath-freshening mouthwash was represented by Scope. Antibacterial mouthwash was represented by Listerine Antiseptic. Fluoridated was represented by Act. Alcohol-free was Crest Pro Health. And finally, the control was commercial saline because it mimicked the saliva within your mouth. Also, the S. mutans bacterium that was tested was purchased from wards. Some additional materials were the inoculating loop, 5 plates, 25 blank discs, tryptic soy agar, incubator, pipettes, autoclave, lab coat, and disposable gloves. For my procedure, each set of blank discs was representative of a different type of mouthwash or control. I had 25 total blank discs, 5 per plate. So all the disc A's were labeled Scope, the disc B's were Listerine Antiseptic, disc C's were labeled Act, disc D, Crest Pro Health, and disc E was saline. All of the blank discs were soaked in their designated mouthwash or control, and then I set up the 5 plates, labeling them 1 through 5. To begin, I filled an empty test tube with sterile distilled water. Next, I sterilized the inoculating loop and placed it into the S. mutans bacteria culture. Then I transferred the bacteria into the distilled water test tube and closed the lid. I then opened one of the 5 plates and streaked the S. mutans thoroughly on the plate using a cotton swab. I repeated this for each of the 5 plates. Then I placed one of each type of labeled disc in each of the plates. I placed the 5 plates containing the discs into the incubator at 37 degrees Celsius for 72 hours and then examined my results. This is a diagram of a simplified version of my experiment. The blank discs were soaked in each of the designated types of mouthwash, then the bacteria was swabbed on each of the 5 plates. The blank discs were placed in their designated spots within the plates, and then the plates were put into the incubator at 37 degrees Celsius. After 72 hours, the zones of inhibition of each disc was measured. Here's a graph of my results. This graph shows the average zones of inhibition and confidence interval for each type of mouthwash. Notice that disc E, which was the commercial saline, was the most effective in killing the S. mutans, while disc B, which was the Listerine Antiseptic, was surprisingly the least. This suggests that rinsing one's mouth with salt and water is more beneficial than using mouthwash. Here is a table of my averages. As you can see, saline had the highest zone of inhibition with 24 millimeters. According to Rollins and Joseph, in order for a substance to be effective, it must contain a zone of inhibition of 16 millimeters or more. This included ACT, Scope, and the commercial saline. Disc D, which was the Crest Pro Health, fell under the intermediate category with a zone of inhibition of 13 millimeters. And finally, Listerine antiseptic was proven ineffective with a zone of 0 millimeters. This is a table of the average price of each type of mouthwash. Notice that the two least effective mouthwashes are actually the highest priced, while Scope, which was actually the most effective mouthwash, has the lowest price. After completing my experiment, I discovered that commercial saline, which was my control, contained benzalkonium chloride. Benzalkonium chloride acted as an antibacterial. Therefore, commercial saline was not an accurate representation of the most effective way to rid the mouth of S. mutans because it was not acting as saliva within the mouth. Therefore, I had to introduce and test 3 new controls. The first new control was 100 milliliters of distilled water mixed with 0.9 grams of table salt. This acted as a pure version of a saltwater solution with no outside variables. Next was 1 teaspoon, 7.2 grams of table salt mixed with 250 milliliters of tap water. This is a doctor's recommended amount of salt water concentration for a mouth cleansing mixture. Lastly, 100 milliliters of distilled water was used as my third control. This was used to compare the difference between regular distilled water and distilled water with table salt. The same procedure was applied as before. The blank discs were soaked in each of the controls. Using sterile technique, S. mutans was swabbed on plates. A blank disc of each control was placed in its labeled position in each plate, and the 4 plates were sealed with tape and placed in an incubator at 37 degrees for 72 hours. This is a table of my averages for the 3 additional trials. As you can see, all 3 controls exhibited no zone of inhibition after the 72 hours within the incubator. This shows that salt and water are not beneficial in ridding the mouth of the S. mutans. There are many factors that affected the accuracy of my experiment. First, the placement of the plates within the incubator varied. In the beginning of the experiment, all of the plates were placed in the incubator upside down in a straight line. However, after the 72 hours permitted to grow, some were pushed further back than others, and many were flipped right side up. Secondly, plate 3 exhibited several growth defects. The bacteria had a more vibrant color and was also much larger. The results for all mouthwashes was significantly lower in plate 3. Also, the amount of mouthwash retained in the blank discs varied. Some soaked longer than others, and therefore it was difficult to tell which discs were more saturated. The bacteria was also not evenly spread. It's possible that some plates, and certain areas within each plate, received more bacteria than others. There was also a strict time constraint to my experiment. The blank discs had to be soaked and placed on the agar in a very short time to prevent them from drying out. Therefore, attention to minuscule detail was often overlooked. Finally, the S. mutans had aged significantly for the 3 additional trials. Because of the snow and the prospect of a nearing competition, fresh cultures of S. mutans could not be ordered. The use of aged cultures was most likely the cause of no zones for the 3 controls. Changes I could make to my experiment would be to find an alternative way to allow the blank discs to dry out evenly. Also, perform the experiment earlier in the week. My experiment was performed on a Friday, and I was therefore not able to check on my plates until the following Monday. If the experiment took place earlier in the week, the plates could be checked each day within the incubator to prevent their placement from being altered. And lastly, for the 3 additional trials, order a fresh culture of S. mutans. For further study, it would be interesting to test other types of mouthwash and see if they're worth the extra money, such as prescription or organic. Secondly, study mouthwash's effects on other types of bacteria within the mouth. The S. mutans only makes up about 60% of the mouth. It'd be interesting to see if different mouthwashes interact with different bacteria. And lastly, test whether benzalkonium chloride could be detrimental to one's health. It'd be interesting to see if the body could build immunity in the oral cavity to the antibacterial. I concluded that Scope is the most effective mouthwash, followed by ACT, Crest Pro Health, and finally Listerine antiseptic. Commercial saline, which was my control, was the most potent overall. However, because it wasn't an accurate representation of saliva, it could not be accounted for within the experiment. And finally, these are the sources that I used in order to complete my experiment. Thank you for listening.